Can you all see my screen?
Yes.
Okay, great. So thanks again for the organizers for inviting me. So as you heard, I'm Miri, I'm a postdoc at the Broad Institute. And we came here about 1.5 years ago. My husband, Aviel, and I, and our 3 children, Moshe, Avigail, and Jonathan, some of you may know. We're very happy to be here and to be part of the Young Israel of Brookline community. And today I will tell you about my research into how tissues maintain their balance. As you heard from Ishai, my background is in physics, but in my graduate studies, I decided to use my skills and my background in physics and mathematics to explore biological systems. And more specifically, my research is focused in systems biology. This is a relatively new field, was founded about 20 years ago. And in the systems biology field, we aim to use a holistic point of view when we explore biological systems. So instead of focusing on specific components, we aim to explore the interaction between different components and how they form a functional unit. And this can be done in different levels. So either at the molecular level, when we explore interaction between different genes that together perform some important function for the cell, or interaction between different cells in the tissue and organs, as I will discuss today, and all the way to interaction between different organisms that form a community. And researchers in the field of systems biology usually use 2 main approaches. The first is to develop mathematical models to describe these interactions between the different components. We use these mathematical models to predict the behavior of a certain biological system, and then we can use experiments to validate those predictions. And the second approach is to develop new computational methods to analyze big data. So, when you think of a typical human cell in our body, it encodes 20,000 different genes, and then you give it time, and we have 30 trillion cells in our body. So we, most of the time, we need to deal with huge amount of data, and we constantly develop new methods to be able to interpret the data. And so today I will show you 2 examples from my own research of how I use a systems biology approach to study tissues. In the first part of my talk, I will discuss how tissues maintain constant proportions of their different cell types in terms of the number of the different cells. And in the second part of my talk, I will discuss how tissues optimally trade off different tasks. And I just want to say that please feel free at any time to stop me if you have questions and something is not clear. Right, so when we think of tissues, tissues are a collection of different cell types that can communicate with each other through different in different means. So for example, different cells can produce signals for each other. These signals, molecules or proteins, can migrate in the tissue or diffuse, or they can also communicate with each other through contact mechanically. And here I just show you a few examples of images of tissues under the microscope. For example, this is the small intestine, this is the kidney and the liver. And if you're not used to stare at these images every day, then I'll just say that here, every dark purple point is a nucleus of a cell, and you can see cells line these very thin walls and form what is called villi in the small intestine. It kind of look like a brush, and it helps to absorb all the food that we eat. And you can see at the bottom different cell types. Here in the kidney, in this image, every red circle is a nucleus, and you can see these columnar cells that form this kind of a circular structure. And in between these circular structure are different types of cells. And here in the liver, in green, you can see the walls of the different cells. And these are liver cells, also called hepatocytes, and you can see how they form this kind of like a beehive structure. So you can see the different tissues have different structure, different cell composition, and as we know, also different functions, but tissues also have common features. And we are very interested in finding these common features. So for example, most tissues are made of what we call a client and an accessory and other accessory cells. And to explain what I mean, it's useful to think of an analogy. So for example, think of a person that specializes in a very special profession. So I like to think of musicians that dedicate their entire lives to playing a certain instrument. And so they don't have the time to perform more basic functions of everyday life. So for example, they need someone else to cook for them, and they would need someone to come and fix things around the house. And they don't even have the time to call these experts. So they need someone like a butler, someone that can coordinate all of the needs in their household. So this is exactly the case in a typical tissue. If you think of a typical tissue, like here I show the intestine, but it's true for most tissues, there will always be a client cell, what we call. These will be the specialized cell. These are usually epithelial cells, and they specialize in a certain function of a tissue. So for example, in the intestine, these are the epithelial cells that line these walls, and they are the experts that absorb all our food. And in our brain, these would be neurons. In our pancreas, these will be beta cells that secrete insulin and so on. So every tissue has its specialist. And then we have other cell types in the tissue that perform more basic functions and can be found in most tissues. So for example, macrophages, these are the type of an immune cell that eat up cellular debris and dead cells. And also these cells are the first responders to any need in the tissue. They are like the butler. We have fibroblasts. These cells produce proteins that are important for the structure of the tissue. These cells are also the ones that produce the scar when we get cuts. And so maybe they're like the plumber. And we have blood vessel cells that form the blood vessels and allow for oxygen and nutrient to enter the tissue. And so in every tissue, of course, there are other cell types, but in most tissues, you can find a version of these 4 cell types. And we also know that tissues are able to maintain constant proportions of these different cell types. Okay, so you need, for example, 20% of that and 30% of the other, and so on. And throughout life, tissues maintain this balance, this constant proportion, despite the fact that there is constantly turnover. So cells, as you know, they divide and they are dying and replaced by new cells. And despite this dynamical turnover, they're able to keep a constant proportion. And we know that it is important to keep this balance because there are diseases that are associated with improper balance in a tissue. So for example, fibrosis is a disease where in response to damage, some cells are removed and are damaged, and they are replaced by a scar. And this also happens to us when we get cuts. And if the injury is mild, then eventually the scar is degraded and cells can regenerate. However, sometimes when the injury is too severe or the cut is deep, we know that scars can persist. And so internally, if a tissue is scarred, it can not function properly and eventually lead to this very deadly disease called fibrosis. And also other diseases such as degenerative diseases and cancer are diseases of where this balance is broken in the tissue. So it's really important for us to understand, how do tissues maintain this balance? How do cells know when to proliferate more and when to balance their division rate and their death rate and to maintain constant levels? So to address this, in my PhD, together with Uri Alon at the Weizmann Institute, I studied how different cell types communicate with each other through producing what are called growth factors. So growth factors are small proteins that signal the cells to divide. So if a cell sends around it a high concentration of these growth factors, it will multiply. If there aren't enough growth factors around a cell, it will not multiply and eventually it will die. And we know that cells can produce these growth factors to one another. And so you can imagine a tissue, let's think of 2 cell types in a tissue, and every pair of these cell types communicate with each other, signal each other to divide. And so we asked, what are the requirements on these interaction between the cells in order to maintain balance, in order to maintain a constant proportions of the cells? Because if they just keep secreting growth factors to each other, they will exponentially grow in their numbers and keep on growing and growing. So something needs to stop this secretion. And by developing a mathematical model that describes these type of interactions, we were able to find a condition, to predict a condition that is necessary for maintaining constant proportions of these cell types. And this condition is that one cell type is close to what we call carrying capacity. This is the maximal number of cells that can be supported in a given environment, for example, due to space availability. So there's just not enough space to grow. But the other cell type is maintained at a constant level because it imposes a negative feedback. So once it senses these growth factors around it, it will consume them, will eat them up, and therefore will decrease their concentration and then will stop divide. And so in a tissue, one cell type will be maintained at some high level due to external conditions like space, as we mentioned. And the other cell will be, the number of these cells will be determined by the availability of the growth factor. So the more of these proteins, these growth factors there is, the more that they will consume them until they will lower the concentration of these growth factors and will stop dividing. And so you can imagine an experiment where you can extract these two types of cells from a mouse, for example, and you can grow them in the lab in a plate. And then you can follow their numbers over time. So for example, you can take a certain level of both of these cells, grow them in a plate, and then after a few days, you can count how many cells you have from each type. And we predict using the mathematical model what would be the behavior of such an experiment. So let's imagine that we can count the number of each cell type. So the prediction of the model is if you start from this point where you have a very low number of both of these cells, the cells will not be able to support each other and they will crash until they will eventually converge to 0 cells. You don't have any cells in your plate. However, if you'll start with a higher level of both of these cells, they will secrete these growth factors to each other, proliferate until they reach a balance where the division rate equals to their death rate, and they will maintain constant levels. And actually, no matter where you start from, even if you start from, you plate different ratios of these two cells, they will end up converging to the same point. And so what determines the fate of the cells is this line. And if you start your experiment from any point around here, any ratio between the cells over here, you know that the cells will divide until they will reach this constant proportion. But if you have a low amount of cells, then they will crash. This type of plot is called a phase plot. This is a very standard way in dynamical systems in mathematics to describe the behavior of a dynamical system. And here we use that to describe the behavior of the cells. And we collaborated with Ruslan Mejitov, who is currently my mentor also from Yale. And his students were able to, for the first time, to actually measure the phase plot. So what you see here are actual experiments. So here are the number of this type of cell, the fibroblast, and these are the number of the other cell type. And you can see every arrow here is an actual experiment where they started the experiment for a certain amount of cells. And after 2 days, they went back and counted the number of cells, and then were able to draw an arrow. And you can see how if you have low number of cells crash and they will die. But if you have enough cells, they will proliferate, they will increase their numbers until they will converge to this point, which is the balance between the cells. Okay, so to summarize this part, I showed you how a relatively simple model for how two cells communicate with each other can help us understand how a typical tissue maintains constant proportions of the different cell types. So at least one of the cell type in a tissue needs to be restricted because of there is no more space or other external conditions, and other cell types will be restricted by the availability of their growth factors. And at the second part of my talk, I want to...
What is the green dot in the...
Sorry, what?
Can you go back to the previous one?
Sure. This one?
Yeah, there's another that is on in the green dot.
Yeah, that's a great question. So actually, interestingly, when these type of cells, they are the fibroblasts, when they are alone, so you can see here is where you have no zero cells of the other cell type. When they are alone, they can produce their own growth factor and they can survive on their own. And so they will converge to this point. However, the other cell type cannot survive on its own and needs the other cell type in order to survive. So this is why there is this asymmetry in this circuit.
Could you go back to the previous slide from this, please, Miri?
Sure.
The one with the negative feedback. Yeah. So did you experiment with modifying or regulating the feedback loop at all to inhibit the growth in any way?
So let me see if I understand the question. You asked if we try to modify this negative
So my
feedback?
understanding is you said that the homeostasis is regulated by the amount of negative feedback that is, received to minimize the new production of cells, right? Mm-hmm. That's what the negative feedback does, is it limits new production of cells, right?
Right, yeah.
So presumably you could, I don't know what that negative feedback consists of, right? Whether it's chemical or what, but somehow you could alter the homeostasis and ratio by modifying the feedback loop, right? As well as just multiplying the cells and seeing what the result is, right?
Right. So currently in our model, in these experiments, this negative feedback is done by a process called endocytosis. This is a process where the cell, once these growth factors binds to the receptor on the cell surface, it internalizes the growth factor with the receptor. And so once it bound, it cannot affect other cells around
it's
it.
like just a limit that's reached, kind
exactly.
of. I see. So it caps it in a sense.
Exactly, yeah.
All right. Well, so that's all right.
Yeah, even if you have 2 times,
sort of a passive feedback in a way.
Right, yeah.
I see. All right, thank you.
I have a question as well. So can you go back to the slide with the green and blue dot? Yeah. So is it possible?
Sorry.
So is it possible that there are more steady state points, but you just didn't look enough?
Right. So from the model, we don't predict additional steady states, but this is, using the interaction that we assumed occur here. But from experiments, well, it's hard to say, but I can tell you that they checked 5,000 fold of ratio between the cells, and they always converge to that point. So you can see this is a logarithmic scale, and even if you really increase the number of scales, like 5,000 times 5,000, it doesn't converge to any other point. So we believe this to be, the only states, at least in this framework.
That's pretty convincing, yeah, okay. Thank you.
Yeah, sure. So, right, so if there are any other questions. So we discussed how tissues maintain a balance in terms of the numbers. And next, I wanted to discuss how do tissues maintain a balance in terms of their function. So, when you think of the different types of cells in your body, you have neuron cells, immune cells, they are very, very different, but they all have the same DNA molecule in the nucleus. And the only thing that makes them different is that they express different genes. So a gene is a short sequence of the DNA, and it is transcribed into an RNA molecule and then translated into a protein. And the proteins are the machines that are used to execute all the cellular functions. And so different cell types in our body produce different set of proteins or different levels of proteins at any given time. So if we look at the proteins or the RNAs that are produced in every cell, it will tell us something about their function. And we have the ability now to measure how many RNA molecules or how many proteins is produced at every cell. And cells from different types, if you can imagine, if you plot, for example, 2 genes and you measure how many RNA molecules are produced for a certain gene at every cell, then you will see different types of cells will lie in different regions in what is called gene expression space. But in fact, every cell is not described only by these 2 genes, right? We know that we have about 20,000 different genes. So every cell is characterized on the gene level by 20,000 numbers, indicating the level of expression of every gene. And this will tell us what this cell is performing, what are the functions that these cells are performing. And when we think of the functions of cells, we know that cells are usually multitaskers. They need to perform multiple functions for the tissue. So here I show an example of the liver, which is really a classical example of the most multitasker cells. So the specialist cell, the musician cells in the liver are called hepatocytes. They need to perform about 500 different functions that are very important. And we know that at any given point, a cell produces a certain set of proteins that are important for specific function. And so no cell can be optimal to all of these 500 functions at once. And so somehow cells need to divide labor. And we want to ask, how do cells optimally divide labor in tissues? So to answer this question, we turn to a theory, it's called Pareto theory, that is from the field of economics and engineering. And this theory tells you if you want to design a system that faces trade-offs, it will tell you how to design it in an optimal way. Okay, so to explain the theory, let's consider, for example, the world of superheroes. Okay, and let's think that we want to design them to be strong and agile. So we can think of example, for example, Spider-Man will have a high level of agility, maybe some level of strength, but maybe it's not the strongest. The Green Hulk, for example, is very strong, but maybe less agile than Spider-Man. And if you think of Pumbaa, then Pumbaa performs worse in both agility and strength.
He's not a superhero.
Exactly. So this is something that the Pareto theory predicts, right? So the theory tells us that Pumbaa will not be selected, to be a superhero because there are others that are better than him in both of these tasks. And so eventually what you're left here with is a line called the Pareto front, which is the set of points that optimally trade off agility and strength. So any point beyond that line is just not feasible. Okay, and below the line, these are points that are suboptimal. And so every superhero here will be optimal, but will trade off these 2 tasks differently. And if we can imagine that there are some traits that we can actually measure for the different superheroes, let's imagine we can measure their height and weight, then this theory predicts that if we have 2 tasks that they trade off, then different superheroes, the optimal superheroes, would fall on a line in this trait space. So for example, the vertices of this line would be what we call archetypes. These will be specialists at each task. Okay, so Spider-Man is the best in agility, and the Green Hulk would be the best at strength. And superheroes in the middle will be what we call generalists. So they are also optimal, but they, for example, they do 50% of that and 50% of the other. Okay, and so if you have 3 tasks that there is trade-off between, you will predict that the individuals in your system would be on inside a triangle. If you have 4 tasks, and tetrahedron, and so on.
So you mean by task, for instance, if another task was special powers, right?
Yeah, exactly.
Okay, thank you.
Yeah, right, exactly. And so going back to cells, for cells, we can measure their height and their weight, but I'm not sure if it will be useful. But what we can measure and will indicate something about their function is their genes, right? So we can measure the different genes that will be equivalent to the weight and height of the superheroes. And we can describe each cell as a point in this gene expression space, telling us how much it expresses of every gene. And you can actually do that, and you can take thousands of liver cells. These are the same cell types, but they are different liver cells from the liver. And if you do that, you see that they will fall in some kind of a cloud in gene expression space. And so we use the Pareto theory to ask whether we can fit this cloud to some kind of a shape, a triangle, a tetrahedron, and so on, to tell us something about maybe the trade-offs that these cells face. And so here I show you, this is real data. Every point here, every blue point is an actual liver cell hepatocyte. And what we did, we fit it to different shapes, and we find that the best fit is a fit to a tetrahedron. And then when you look at cells that are closest to the vertices, these will be the archetype, these will be the specialists. So by looking at what genes they express, what proteins they produce, we can identify the 4 tasks that they specialize in. And we found 4 complexes. So this is a short list for every function. We found 4 complexes of liver functions that these cells trade off. And so you can implement this theory for other cell types, other tissues, and understand trade-offs that cells face and how they solve it.
So these are the musician cells, right? These are the top, the epi, whatever.
Right, the specialist, yeah.
All right.
Yeah.
So, all right. So each one of those is a multi-instrumentalist in a sense, right?
Right.
Okay, thank you.
Right.
Are you measuring these in vivo or in vitro?
So this is in vivo. This is a single cell RNA sequencing.
And they're in the actual tissues and in petri dishes.
Yes.
Oh, okay.
Yes. This is a mouse. This data, mouse.
And the expression are normalized, right?
So the expression is normalized. It's and we use methods to lower the dimensionality. These are not the actual 3 genes, but it's a projection of the 20,000 dimension onto the first 3 main axes, principal components.
Why is there one dot that's outside one of the vertices or whatever? Yeah.
So we do allow for cells to be also outside. We look for, so the way we do that currently, we measure the convex hull. So this is the shape that encompasses the cells. And then we find the shape that the volume will best fit this convex hull.
It's an approximation.
Yes, yes.
All right,
so
thank
as you can see here, the ellipsoid actually indicate the arrow of the position of the vertices.
Yeah, which is not exactly fixed. All right.
Exactly.
Thank you.
Sure.
Wait, so each axis is not one gene, but a linear combination of genes.
Exactly. Yes.
And were you able to find the commonality between those axes?
Yeah. So yeah, they have some common feature. They have some meaning, and yeah. Did that answer? I don't know.
Yes, you didn't tell me what the commonality, but yeah.
So they go in line with these tasks, of course. Some combination of these 4 different tasks. So the first axis is mainly secretion of plasma proteins. For example, albumin is one of the most important proteins we have in the blood, and the liver is the only organ that produces that. So that's a very important function.
Great, thank you, Miri.
But once you remove these cells, haven't you perturbed their behavior? So therefore, by the time you measure it, it might have changed?
Well, it could be. So I don't think that's the case. We have methods to remove cells that we think are in stress. For example, if they have high mitochondria genes level. So yeah, I don't think this is the case. And also, we collaborated with the lab at the Weizmann Institute that they also did experiments in situ. So they were actually able to color the expression of different genes in the tissue without, causing any stress to them. And you can actually see how the different genes in every archetype are expressed in different cells and also in different locations in the tissue. So we believe that the cells that are in different, cells in different archetypes would be cells in spatial position where the condition is best for their function that they're performing. So for example, cells that need to produce proteins will be in an environment that is very rich with oxygen. But cells that need to detoxify, actually, they don't need oxygen. So they would be at positions where level of oxygen is low in the liver. So actually, this is my last slide. I hope I showed you how combining theory and experiments can be very useful to understand biology in general and of tissues in particular, and how tissues balance their numbers and their function. And if you have more questions, I'll be happy to answer.
Thanks. I just want to say that was really fascinating. I was just trying to think about what kind of applications you could use this for, and what, you know, maybe as you're furthering your research, what are you looking into next?
Thank you. So yeah, in terms of applications, I think some of the things I showed you can be very important in disease states. So for example, going back to the Pareto theory, we are looking at tumor cells, cancer cells, and if you can understand the trade-off that they face and what they specialize in, and we think maybe we can target better than with specific drugs that will target their specialty. So for example, if your tumor cell is very, very important in producing a certain protein, so you can try to damage that function, and then, you know, and there are some experiments that we've seen that it can be very useful. So this is one example. With the model that I showed you with the first part of my talk with the cell circuits, I mentioned fibrosis, and actually we do have like a extension of this model of the behavior of cells in fibrosis in response to damage. And we think we might have insights about what type of drugs or targets to produce to prevent fibrosis. So by the way, fibrosis is a disease that can occur in all tissues. For example, in the liver, 30% of fibrosis diseases end up in liver cancer. So it's really horrible disease, and there is no cure currently. So yeah, I hope that this will be useful for that.
How does cell senescence, meaning that cells have a limited number of divisions before they sort of die permanently, how does that factor into all of this?
Yeah, so senescence is really interesting. And actually, it seems that there is like a paradoxical effect of senescence. So when we are young, I think senescence is kind of a useful way for the cells to stop dividing. And so, for example, in response to injury, the cells that produce the scar, they become senescent and they stop being activated. And so there are less scars when we are young. But as we get older, more cells become senescent. And then the bad side of senescence is that they keep producing inflammatory signals. And so when we are older, we have always this kind of a basal level of inflammation. Some people call this inflammaging. So you have always like a low level of inflammation, and this chronic inflammation is actually a driver for fibrosis and for cancer diseases. And so, yeah, so senescence really has a paradoxical role. And yeah.
Would this have any, with this cell senescence, would it be able to be manipulated in any way so that, for example, that keloid formation could be decreased?
So I think there are a lot of efforts to try and do that. For example, you can activate the immune cells to clear these cells and to lower the chronic inflammation. So there are some efforts to do that. And yeah, it's there's a lot of research going on with senescence.
Miri, it sounds like one of the earlier slides where you described a disease model, it sounded like maybe even for something like cancer, it wasn't really particular to the individual cell. Like the layman's understanding, it's a rogue cell that multiplies, but it's actually more endemic to the tissue, to a cluster of cells, really, and the ability of the tissue to regulate itself rather than the individual cell.
So, right. So with cancer, if you know, from my understanding, so cancer cells, they are mutated, and so they cannot, they don't respond properly to the level of these growth factors. They just, you know, go wild, and they don't care how many growth factors there are. But we do know now currently that what is called the tumor microenvironment, so the environment of the tumor cells, like for example, let's say liver cells, these hepatocytes become cancerous, but we now have evidence that the fibroblasts, the macrophages, all the cells around them, like the Butler cells, and they also become very, very strange. Their behavior is very strange, and they're like changing the behavior to support the cancer cells. So you're absolutely right that now there are a lot of research going on to understand the supporting cells, and these circuits can be very useful for that. We think that something goes wrong in their behavior, in the way that they produce these growth factors. And if you target these cells, you might...
Kind of like Trump's cabinet, if you'll forgive the analogy, everyone, right? Sorry. Sorry.
Maybe I'll stop share that we can see each other better. Yeah.
Miri, I have a question. Is there any similar experiment done with neurons or brain cells?
Do you mean for the first part or the second?
I thought of the second, but maybe also for the first.
To understand their trade-offs, you mean?
Yeah, and maybe to look for some kind of Alzheimer's disease medication that might influence.
Yeah, definitely. So first of all, I've been looking I've looked at several data sets of neurons, and I do think that there's a lot of heterogeneity there, and this theory can be really useful there. And currently I'm looking at cells in the retina. And yeah, so definitely. And if you want, we can talk more. And, if you have a certain, specific data set in mind, and yeah. There's a question in the chat.
Do you find any difference between the working here in America or in Israel, the culture of work?
Wow, what a question. Yeah, of course there are differences in the culture, at least. Although I have to say, so I grew up in Weizmann Institute, and the Weizmann Institute is very, very, you really feel very international there. So I don't know if you know, but all the courses are done in English. And so and there are a lot of students and postdocs that come from Europe and the States. And so like in the past 10 years, I used to speak in English every day. So maybe for that, I don't have the good, I don't have the typical culture of an Israeli lab. And other than that, I think labs here tend to be bigger in terms of the size of at least in biology. I don't know, maybe that's not true. Yeah, but the science, I think, is great also in Israel. And so as you can see, I mean, of course, the Harvard and MIT and the Broad Institute are amazing places, and there's new technology, but postdocs that come here keep bringing all these new technology also to Israel. So there is really good science done in Israel too. So this is my experience.
Yeah, let me read the chat and explain. I specialize in dealing with complex patients, and one of the challenges is to deal with patients with multiple comorbidities in which the ability of one problem and its solution, that is the medication, can affect others. We have very poor formal ways of addressing these patients. I wondered if the mapping that you discussed before can actually be broadened to allow us to develop a formal methodology to define a patient as not as one of many people, diabetes, let's say, which has multiple subgroups, and typically problems with cholesterol, but to actually be able to define them within some formal method such as yours.
Yeah, that's a really interesting point.
The problem, as everyone knows, in medicine is there's fragmentation, and there's a specialist after specialist, and even now, for instance, there are 5 different well-defined specialties of cardiology, which itself is a specialty. That makes thinking holistically very difficult. And so we do certain things, we make lists of problems and so forth, but that's totally intuitive as to how they interact. This seems to be a formal way of defining where on this diagram the patient lies, and allow us to also monitor them as the various problems change in intensity and so on.
Maybe, doctor, also that patients are more complex than cells and have more multiple functions and roles than than any individual cells do, as well as stimuli. So it's much harder to control for that, even define what the concerns are, I would imagine.
Well, I think that we're moving to the point where, in fact, we've got an ability to define individual diseases very precisely, particularly with genetics, as an example, and other ways. But we have no way of linking this. And so I don't have the mathematics background to address this, but it seems to me that the method is not dissimilar. It's complex, and that's correct. We're very complex, but we need to start somewhere in this.
Especially environmental factors, which is a petri dish with a cell. It's real life with patients,
Yes, I know.
Thank you.
I think one way of thinking of complex patients, and I'm an anesthesiologist, but I think I do this every day. And you think about, what are you trading off, right? So you can picture that patient in the tetrahedron that, Miri showed in the slide, and they're going to be at one place in that tetrahedron, right? And that patient, you want to optimize their medications, what you're, how you're treating them to, sort of avoid side effects in one area and try to get the benefits of those meds in the other area. And I think we just kind of do that inherently, but I think it's fascinating what you're suggesting to really think about the mathematics behind this. And maybe there's a way to optimize how we're prescribing meds, and how, maybe it's best to lower doses of certain things and, increase doses of other things when you factor in multiple comorbidities. I think that's a really interesting
absolutely on
that you're raising. Yeah.
Absolutely a target. So before I decide to introduce a new medication to this patient, I look and this will allow me to look appropriately at its effect on other tissues that are now, or even normal tissues, and how their functional may be moved one way or another, and address and add new problems or solve new problems.
Yeah. I would just want to say, I agree that it will be really interesting to think of, in terms of this problem. Just want to say that we tested this theory, the Pareto theory, not only on cells, but on organisms, different organisms. We do see that you can just give really interesting insights. For example, in looking at ants in a colony. So, you know, ants are like fighter ants and ants that are foraging and nurturing, and you really see them line really beautifully in a triangle, depending on some features of the ants, the width of the head, or some chemical that they produce. So it definitely can be also at the level of organisms.
This would be worth a theoretical paper, I think, in some of the medical journal, because there is really no methodologies that are formal for doing this. So as Sarah said, this is intuitive, and the fact that we do as well as do is very nice, but there's obviously anybody who does this recognizes that it's a hit and miss, trial and error type of function, not.
Can you do this with higher dimensionalities, you know, more than just 3 factors?
Yeah, sure. So you mean if we can fit to more than 4 tasks?
Well, yeah, well, in that graph, you have 3 different genes.
Yeah, so so these are not really, so every axis is not a gene, it's a combination, linear combination of all the 20,000 genes in the data. Oh, so it's like we use methods to look at the low dimensional manifold.
Okay.
Could I ask you, I mean, you looked at individual types of tissue like liver tissue, kidney tissue. What about the whole body? Sort of what Dr. Levinson was getting at, but at a more granular level. Suppose all the butlers like have the same skills and training, right? That it's really only the top level cells that are different. And how much communication is there between tissue manufacturers and tissue helpers to create a whole environment for all the tissues to function syncretically, right? That's what a body is, an organism.
Yeah, definitely. So there are some examples in the body where there is a crosstalk between different organs. So for example, the gut and the brain and immune system, blood, there's a lot of crosstalk over there, and it's fascinating to explore. But what you mentioned is actually something that I'm now exploring. One of the things that I'm doing, I'm looking at all the cells from all the body, all the tissues, from brain to skin. And I'm trying to find the genes that all the cells express, right? So these will be like some very core group of genes that all cells need to express just because they're cells. So these are very, very basic functions. We call them, in biology, we call them housekeeping functions. So these are housekeeping that everyone needs to do. Musicians need to do them, doctors, rabbis, everyone needs to do them. And then we ask, what would we see? Do we still see that some cells, depending on their tissue and depending on their specific function, would they perform these housekeeping functions differently? Would they adjust? And then
the rabbi schedule is different from the musician schedule.
Exactly.
Different hours, presumably.
Right. And what would determine that? Would the tissue of origin determine that? Or would the fact that they are immune cells versus muscle cells versus...
Interesting. Wow. Yeah, boy, so. Universes. Wow. Amazing.
If there are no more questions, thank you, Miri. It was very fascinating.
Thank you.
And if anyone wants to share his thought, his or her specialties or hobbies, you're welcome to approach me. And have a good weekend.
Shavua Tov, everyone. So, good to see you all.
Yeah.
Be well. Thank you, Dr. Adler.
Thank you.
Bye.
Bye-bye.
Bye.